Welcome to Principles of Ecology. Today we are going to explore a fascinating and fundamental concept in biology, the difference between unitary and modular populations. When we look out at nature, we often assume that counting individuals is a simple task. But as we will soon discover, the way an organism grows completely changes how we must study it. Understanding this distinction is absolutely critical for ecologists, and by the end of this video, you will see the natural world in a completely new way. Let us dive in. To begin our journey, imagine you are trying to count the number of dogs in a park versus the number of grass plants in a lawn. Counting the dogs is incredibly easy. Why? Because each dog is a distinct, separate individual with a clear beginning and end. A dog grows to a specific size and then completely stops growing in its physical form. However, if you look at the grass, it is incredibly confusing. Many blades of grass are actually connected underground by horizontal stems. The grass just keeps growing, adding new blades and roots indefinitely. It becomes nearly impossible to tell where one plant ends and another begins. This fundamental difference in growth is the core of our topic today. Now that we have an intuitive grasp, let us formally define these categories, starting with unitary organisms. In ecology, a unitary organism begins its life as a single zygote, which is simply a fertilized egg. From this single starting point, it grows into a highly predictable, determinate form. For example, a human will always develop 2 arms, 2 legs, and 1 head. More importantly, unitary organisms have a strict limit on their growth. They will eventually stop growing once maturity is reached. Because their physical form is so strictly defined by their genetics, it is very easy for ecologists to identify and count them in the wild. In stark contrast to unitary organisms, let us look at how modular organisms develop. A modular organism also begins as a single zygote, but instead of growing into a single fixed body, it develops into a basic unit of construction called a module. This initial module then repeatedly copies itself to form a branching, indeterminate structure. They do not have a fixed final shape or size. Trees, corals, and sponges are classic examples of modular organisms because they continuously add new branches, polyps, or shoots throughout their entire lives. This continuous, repeating growth pattern allows them to expand indefinitely as long as environmental conditions remain favorable. Because modular organisms grow by copying themselves, we must introduce 2 critical terms to avoid confusion: genets and ramets. We must carefully distinguish between a genet, which is the entire genetic individual originating from one single zygote, and a ramet, which is a single, potentially independent module or shoot of that genet. Think of the genet as the genetic parent, and the ramets as the physical clones it produces. A single genet might look like a whole forest of trees above ground, but because they all share the same root system and DNA, they are technically just one genetic individual made of many ramets. Understanding this terminology brings us to why this distinction is absolutely critical for ecologists in the real world. When ecologists want to know if a population is thriving or dying, they measure population size, density, and growth rates. For unitary organisms, like bears, counting is straightforward because one physical body equals one unique genetic individual. But for modular organisms, like a field of aspen trees, A forest of thousands of tree trunks might actually be just one single genetic individual connected underground. If we do not account for this modular growth, we might falsely conclude that a population has high genetic diversity, when in reality, it is highly vulnerable to a single disease. To avoid these ecological errors, we use specific mathematical models, starting with the formula for unitary population density. Because every physical body of a unitary organism represents one unique genetic individual, we can directly use the standard population density formula. The formula is D equals N divided by A. Here, D represents the population density, which tells us how crowded the habitat is. N stands for the total number of individual organisms we counted in the field. Finally, A represents the total area of the habitat, usually measured in square kilometers or hectares. If the number of individuals, N, increases, while the area, A, stays the same, the density, D, will increase, meaning the animals are more crowded. Now that we have our formula, let us see this with a concrete example involving a unitary population. Suppose a wildlife biologist is studying red foxes in a protected forest reserve. Through careful tagging, the biologist counts exactly 150 individual foxes. The reserve has a total area of 50 square kilometers. We want to find the population density. We substitute our known field measurements directly into the density equation. We place the 150 foxes into the numerator and the 50 square kilometers into the denominator. Performing the basic division of 150 by 50 gives us 3. This means that on average, you would expect to find 3 distinct, genetically unique foxes in every square kilometer of the forest. Having seen how simple unitary populations are, let us look at how we mathematically represent modular populations. For modular organisms, a simple count of bodies is not enough. We must calculate the total ecological footprint by looking at both the genetic individuals and their cloned parts. The formula is N total equals Ng times R average. In this equation, N total represents the total number of observable physical units, which we call ramets, in a given area. Ng stands for the number of genets, which are the unique genetic individuals that originally grew from a seed. Finally, R average represents the average number of ramets produced by each genet. If the environment becomes highly favorable, the value of R average will increase rapidly as the plant sends out more shoots, causing the total physical population to spike. Let us apply this modular formula to a real-world scenario. A botanist is studying a modular population of wild strawberries in a meadow. Through genetic testing, the botanist determines there are 12 distinct genetic individuals, or genets. On average, each genet has produced 45 connected shoots, or ramets. We need to find the total number of physical strawberry plants. We begin by plugging the 12 unique genets into our equation. We then multiply this base number by the 45 ramets that each genet produces on average. Executing the multiplication of 12 and 45 yields a final numerical value of 540. This tells the botanist there are 540 distinct plants actively photosynthesizing, even though they all stem from just 12 original seeds. To truly cement these concepts, let us look at a visual comparison of unitary and modular organisms. On the left side of the diagram, you see a mouse. This represents our unitary organism. Notice the label pointing to it. One individual equals one genet. The physical body and the genetic identity are exactly the same thing. Now, look at the right side, where we see a strawberry plant.
This represents our modular organism. Notice how the horizontal runners connect multiple leafy shoots together. The large bracket at the top shows that this entire connected network is just one genus, originating from a single seed. However, the arrows point to the 4 distinct physical shoots, labeling them as 4 ramets. This perfectly illustrates how modular organisms conquer space through cloning.
We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that unitary organisms have a fixed developmental path, making it easy to equate one physical body with one genetic individual. Second, we discovered that modular organisms grow by continuously adding repeated parts, forcing us to look at populations differently. Third, and most importantly, we must always distinguish between the genetic individual, known as the genet, and the physical modules, known as ramets. Mastering this concept ensures that when you measure population size, density, or genetic diversity, you are counting the right units for the right ecological questions. You now have a solid foundation in understanding unitary and modular populations.